Hello, dear students. I sincerely hope that you are feeling good at your homes with your family and friends. And you must be enjoying your studies and welcome you back onto the microbiology class. As you all know that we were discussing one of the important family in the medical microbiology. And the name of that family is the Enterobacteriaceae. And as we all know that we have almost more than 20 clinically significant genera in this family. And for the sake of discussion and for the easiest or the convenience of the discussion, these genera, they have been subclassified. They have been grouped into three main groups with reference to their appearance in the anatomical sites. We have some of the microorganisms known from different genera that are pathogens both within and outside the enteric tract. There are some pathogens which are primarily within the enteric tract, and there are some pathogens which are outside the enteric tract. We are discussing the organisms which are present outside the enteric tract, and we have already gone through what you call it as the Klebsiella Enterobacter, and we are through with the Proteus-Providencia. We have already with that of the other members of the genus. We will now move on to another important organism, the group of microorganisms, what we call as the Pseudomonas aeruginosa. Now, this is, as you all know, that most of the members of this family, they are gram-negative rods, and like others, member of this family. The Pseudomonas aeruginosa, it causes diseases that may vary from sepsis to pneumonia and the urinary tract infections, particularly in patients who have lower force defense. The lower respiratory tract infections, particularly in the individuals who are having this cystic fibrosis. Now, cystic fibrosis is a genetic disease which is progressive in nature, and it is caused by, it causes the persistent long infection, and it limits the ability of the lungs to breathe for a long period of time. Now, this disease, the cystic fibrosis, is caused by mutation in the gene, and the gene responsible for this mutation, it causes or it controls the flow of the salt and the fluids in the blood in and out from the cell. So there is a mucus secretions and become sticky and become thick, and that leads to the some disturbances in the respiratory tract and the difficulty in the breathing. So this organism is primarily responsible for the lower respiratory tract infections, particularly in individuals suffering from genetic disorders of cystic fibrosis. This organism is again one of the major causes of wound infections, particularly the burn wound. They are being contaminated with this infection, and it also causes malignant otitis media, particularly in diabetic individuals. It is one of the most important causes of ventilator-associated pneumonia. So if you look at this, I mean, the first two lines of this, the organism is responsible for a couple of diseases that may cause localized infection, or it can go to the systemic infection. This means that Pseudomonas aeruginosa is fully equipped with all weapons that are necessary for its pathogenesis to cause signs and symptoms in the local or the systematic infection. Now we have 2, 3, 4 important species of the Pseudomonas, and the Pseudomonas, it has been reclassified, or you can say it has been renamed. And the Pseudomonas aeruginosa, it has been now renamed as Bacteroides Prevotella. So because of some changes into the genetic makeup, this organism has been renamed as, it has been redesignated as Bacteroides Prevotella. So whenever you come across a question, or you come across a genus, or come across a species in the questions paper, or somewhere in the diagnostic lab reports, the Pseudomonas aeruginosa or Bacteroides aeruginosa, they're one of the same name given to the same organism. We have another organism in the Pseudomonas genus, what we call as the Pseudomonas specia, and it has been renamed as Bacteroides specia. This means that we can say that out of these 4 species of Pseudomonas, the 2 has been renamed as Bacteria. So Pseudomonas aeruginosa renamed as Bacteria aeruginosa, Pseudomonas specia renamed as Bacteria specia, and the Pseudomonas multifila, it has been renamed as Xanthomonas multifila. And it has been rather renamed again into what you call as the Pseudomonas multifila. So there's one other same thing. Another organism belongs to the same genus, and it causes disease in equines, in horses, and mules, and donkeys. The name of the disease is melioidosis, and it is caused by organism known as Pseudomonas pseudomeliae. So we, the Pseudomonas aeruginosa, Pseudomonas specia, Pseudomonas macrophylla, and Pseudomonas pseudomeliae, they have been renamed. 2 of these species belong to now the Bacteria, 1 belongs to Xanthomonas, and the 4th one remains the same. So we can see that the organism is responsible for a quite variety of infections in humans, starting from, you see, the lower respiratory tract infection, wound infections, particularly in the males, otitis media, particularly in the diabetic, ventricular associated pneumonia, particularly in the hospitalized individual, UTI infections, particularly in those individuals who have the low host defense, and sepsis and pneumonia. Now, if you look at the important properties of the Pseudomonas aeruginosa, as we all know that the organism is a gram-negative horse, and it has resembled to the other members of the family Pteropacteriaceae. It has strict aerobe. This is a unique point with reference to this Pseudomonas aeruginosa. Most of the members of the family Pteropacteriaceae, like Salmonella, Shigella, Escherichia, Proteus, Helicobacter, Campylobacter, they are facultative anaerobes. But this organism is strict aerobes, and because of this anaerobic characteristic, it derives this energy from the oxidation, not from the fermentation. It does not ferment the glucose, and it is one of the non-fermenters organism. The organism is oxidase positive, and it is able to grow. It can grow in the water-containing traces of the nutrient. I mean, the tape water, which is not much rich in the nutrition for the growth of the microorganism. But the Pseudomonas aeruginosa can grow in water, which is having very limited amount of nutrient, and it favors their persistence, particularly in the hospitalized environment. So this organism is one of the most important organism that can cause nosocomial infection. The organism found can grow in the what you call the hexachlorophene, which is a component of the soap solutions, the antiseptics, and detergents. So washing with soaps or using detergents for the cleanings of the hospital equipment or using soap, it does not really have much effect on the survival of this organism. The organism has the potential to grow even in the soap solutions, antiseptic, and detergents. And this makes the basis for the wide spectrum antimicrobial drug resistance of this microorganism. The organism is very much resistant to many chemicals used in the hospital environment for the infection control. This organism, it produces 2 kinds of pigments. When we grow this microorganism, this organism causes infection somewhere, particularly in the skin and the burn wounds. It produces 2 types of pigments. One is the pyocenon. It gives, you see, the blue color to the wound or to the pus. And the other one is the pyoverdin, that is fluorescence in nature. It fluoresces. It is a yellow-green pigment that fluoresces under the UV lights. So it helps in the detection of skin infection in burn patient. If you have a burn patient, and it has been infected with the Pseudomonas species, and if you put some fluorescent, you see, light on it, you can see that there is some yellow-green pigment appearing on that. These pigments, they diffuse into the agar when we culture this microorganism onto the agar plates, and it imparts the blue-green color to the agars also. So you see, this is because of the presence of the pigment pyocenon and pyrovoidin. Now, these pigments, they also contribute towards the virulence of the microorganism. We will discuss the pathogenesis. I will let you know that they have an important role to play in the pathogenesis of this organism. The Pseudomonas aeruginosa, it synthesizes only pyocyanin. Only pyocyanin synthesizing species is the Pseudomonas aeruginosa. The Pseudomonas aeruginosa, it causes, as we have seen, the cystic fibrosis, and because it has the glycocalyx. Under the glycocalyx has the slime layer, it, as we all know, it facilitates or it mediates the adherence of the organism to the mucous membrane of the respiratory tract, and thereby it prevents the antibody from binding with the cell. So this will contribute towards the virulence of the microbe. The organism has a glycocalyx that helps in the adherence to the microorganism with the mucous membrane and prevent the antibody from binding with this microorganism. The organism is found in the soil and water. 10% of the individuals, they carry as a normal flora of the colon. The most skin, the moist skin, upper respiratory tract of the hospitalized patient. So the organism is commensal in the colon, and the moist skin areas, in the upper respiratory tract of the hospitalized individuals, it is very common. That's why it is associated with the ventilator-associated pneumonia. It can grow in aqueous solutions, resulting in contamination of respiratory therapy. So the organism can grow in Texas solution that are used for the respiratory therapy instruments, anesthesia equipment, intravenous fluids, distilled water. So these fluids, like the intravenous fluids, the distilled water, are the fluids used in the anesthesia equipment, respiratory therapy equipment, they can be contaminated with this microorganism, and Pseudomonas aeruginosa can easily grow in these fluids. It is a persistent pathogen with extensive burns, chronic respiratory diseases, what we call it cystic fibrosis. In immunosuppressed individual, it is one of the persistent pathogens again. And all those individuals who are neutropenic, they have neutrophil count less than 500 per liter. And it is also suited with the indwelling catheters. So you can very well imagine that an individual, if he is hospitalized or the patient is hospitalized, undergoing kind of surgery, having you see the indwelling catheters, having you see it has the suppressed immune system, it has burns. So these kind of individuals, they are more prone to this infection with Pseudomonas. Very notorious microorganism in the hospital setup. 10 to 20% of the hospital-acquired infection, they are caused by the gram-negative nosocomial pneumonia. This organism is responsible for the gram-negative pneumonia in the hospitalized setup, and it contributes at least 10 to 20% of the total cases of pneumonia. Total cases of pneumonia in hospitalized setup caused by gram-negative organism, this Pseudomonas aeruginosa has its share of about 10 to 20%. So one of the major causes of hospital-acquired gram-negative pneumonia. The multiple virulence factors that contribute towards this virulence, there may be endotoxins, it can form the exotoxin, there are various enzymes. The exotoxin, they cause necrosis, and they have the enzymes like the elastase, protease, and they causes the invasion of the microorganism in the bloodstream. The pyocyanin, it damages the cilia and the mucosal cells of the respiratory tract. And the Pseudomonas aeruginosa type C secretion system is significantly more virulent and transfers of toxins from bacterium directly into the human cells. This means that the organism is not that much invasive, but the toxins, it avoid the neutralizing antibodies and directly damage the surrounding cell area. UTI infections, you see the clinical findings causes UTI infections. We're already with that in the previous slides. It causes pneumonia, particularly in the cystic fibrosis individuals, and wound infections, especially in the burns and the sepsis. There is black necrotic lesions, what we call as the ecthema garinosum. There is sepsis, and the mortality in this case is very high. The endocarditis, particularly in the intravenous drug users, and there is what we call the malignant otitis media, otitis externa also. The skin lesions, particularly the folliculitis in the users of swimming pool and hot tub waters, it's very common. The corneal infections in contact lenses has also been reported with the Pseudomonas. So wherever there is a fluid, there is a water, there is a moist area, even that it is not that much rich in the nutrients, this organism has the ability to survive and to cause infections. The most common cause of osteochondritis of foot in the particularly the sustained punctures wound to the sole of the gym shoes and all that. Now here you can see the cellulitis caused by Pseudomonas aeruginosa, and if you look at at this side, we can see that there is infection in the burns. So you can really imagine the yellow and the green pigmentation, and the fruity smell is very characteristic. So based on to the appearance of the exudate and the wound area, whether it's a light yellow in color, greenish, or some bluish in color, and the fruity fragment, the fruity odor is really characteristic to identify the microorganism. All right, we can grow this microorganism in the clinical laboratory for the diagnostic purpose. It's a non-lactose fermenters. Colonies, they are produced in the McConkie agar or onto the Eucin methylene blue agar. The metallic sheen of the growth on the TSA agar. Blue-green pigmentation due to the pyocyanin and pyowarden on the agar. This fruity aroma. You smell like the dirty fruits, and sufficient to make a presumptive diagnosis. So we can presume the presence of this microorganism if we grow it on the culture on the agar plate, and we see blue-green pigments on the surface of the agar. The organism can be confirmed by the biochemical reaction. It is oxidase. Sorry for the nuclear. Now, this is how the colonies of Pseudomonas aeruginosa they appear onto the agar. You can see the real greenish pigmentations, along with to some extent the yellow color. So this is a characteristic of the Pseudomonas colonies. We can also see the colonies onto the other medium. So the coloration of the colonies, along with that of the fruity odor of the area involved in the infection, we can really diagnose the Pseudomonas aeruginosa organism. The treatment for this microorganism, the organism is very much resistant to many antibiotics. Rather, it's notorious for its resistance. And the treatment must be tailored based on to the sensitivity of the isolates, and we should be frequently monitored the efficacy of the drug, because the drug resistance can emerge during therapy even. The organism is very much resistant, so the anti-Pseudomonas penicillins like ticarcillin, piperacillin, plus the aminoglycosides like gentamicin or amikacin, they are the drug of choice. The infection caused by the Spacia and Multifila, the drug of choice is the trimethoprim and sulfamethoxazole. We can adopt some of the measures to prevent these infections, and we will have to keep the neutrophilic count more than 500 per liter, because the neutropenia has been shown to be one of the predisposing factors for this infection. If it is because of if you are using some of the indoubling catheters, we should have to remove them as early as possible. And the special care of the burn skin must be taken so that we can avoid the further spread of this organism. Other measures to limit the infection in immunocompromised individuals can also be adopted. That was all about the Pseudomonas, and now we enter into another group of microorganisms that cause infection outside the respiratory tract, and these are the Proteus-Providencia-Morganella Group. The disease caused by this group are the UTI infections, and this UTI infections can occur both in the community and the hospital setup. So the person can have a hospital-acquired UTI infections or maybe in the community. The important properties of the group of this microorganism, they again are the gram-negative rods, and they produce a different kind of phenylalanine demyelinase enzyme. This organism is a group of this microorganism. They are urease positive. This means that they can convert the urea into ammonia and the carbon dioxide. The organism, particularly the first one, the Proteus, they're very motile, and they produce very striking swarming effect on the blood agar plates. And this characteristics, expanding rings over the agar surface appears. By isolating the organism or by culturing the sample on the agar plate, if we observe a swarming growth on the agar plate, like expanding rings over the surface, this is almost characteristics of the Proteus infections or infection by this group of microorganisms. The organism, they have the O antigens of the Proteus, and specifically the OX2, OX19, and OXK. These antigens, they cross-react with the antigen of the rickettsia. So this is what we call as the wheel-flex reaction. In order to identify the rickettsia, we use the Proteus antigen, and if the body responds positive to the Proteus antigen, we think that the rickettsial organism are there, and the name of that test is the wheel-flex reaction. There are 4 medically important species of the Proteus, but based on to the molecular analysis, the 2 of these 4 significantly involved species has been differentiated. With the, like we have seen in the case of Pseudomonas, the Pseudomonas aeruginosa has been renamed as Bacteroides Prevotella. Similarly, the Proteus species, they have been renamed as, like the Proteus-Morganella has been renamed as now Morganella morgani. The Proteus recri now has been renamed as the Providencia recri. Now, these organisms, they can be distinguished from Proteus vulgaris and Proteus mirabilis based on to these several biochemical tests. So, as we have seen in case of Pseudomonas, same is the case in case of Proteus. Some of the species of the Proteus, they have been renamed or redesignated as Proteus instead of, you see, we have now Morganella. And as Proteus, instead of Proteus, we have the Providencia recri. And further differentiation between the Morganella, Providencia, and Proteus vulgaris and Proteus mirabilis can be made based on to the chemical test, biochemical test. Now the pathogenicity of epidemiology of this group of microorganisms, these organisms, they are present in the colon, the soil, the water, and they have the tendency to cause the UTI infection. Now, as a general rule, all those microorganisms which are present in the colon, they have the tendency to cause UTI infection, particularly in the female, where the proximity of the UTI is there. Now, there's vigorous motility of the Proteus, and this vigorous motility of the Proteus, it has a great contribution towards the union of the microorganism in the case of UTI infections. The organism is urease positive, strongly urease positive, and it hydrolyzes the urea and converts into the ammonia and carbon dioxide, and that rises the pH. And the alkaline urine, it encourages the, what you call it, the caliculi, I'm sorry for that, the caliculi. And the caliculi that has produced or made as a result of alkaline pH in the urine because of the action of the urease, which is present in the Proteus organism, is known as struvate. So struvates are the caliculi which are found as a result of hydrolysis of the urease enzyme present in the Proteus organism. Magnesium ammonium phosphate is the composition of these kind of caliculi. These caliculi are the stones. They obstruct the urine flow, and thereby they damage the urinary epithelium. And this area, it serves as a focus point or as a nidus for the recurrence infections. The organism is there, it has become lodged over there, and it can cause the recurrent infection by trapping the bacteria within the stone. So the stone formation result into the damage of the epithelial cells, and this damaged area becomes the focus for the recurrent infection by trapping the bacteria within the stone. Now, the alkaline urine, it favors the growth of the microorganism and the more extensive renal damage. So what we have to do, we will have to keep the urine at a low pH. So the alkaline urine, it favors the calculi formation. It favors the multiplication of the microorganism. So we will have to lower the pH of the urine. Right. The clinical findings, almost the signs of UTI, and it cannot be really distinguished from the UTI infection caused by the Escherichia coli or other members of the family Enterobacteriaceae. The Proteus species, they can also cause the pneumonia. They are involved in the wound infections. They can lead to even septicemia. Proteus mirabilis causes you see the hospital-acquired infections and the community setup infections. And the Proteus gary, it emerges as agent of the nosocomial infection. So both species of the Proteus mirabilis and gary, they are nosocomial infections organism, whereas the mirabilis, it can also be involved some of the community infections. The laboratory diagnosis is based on to the highly motile nature of this microorganism. This organism, when cultured on the agar plate, it produces swarming growth, and swarming overgrowth, it occurs in the blood agars. And if we want to really have the isolated colonies for this microorganism, for that purpose, we will have to add phenylethyl alcohol in the medium, which will inhibit some of the growth of this microorganism, and you can have the isolated colonies. Otherwise, you will see the swarming grow, and we cannot pick the isolated colonies, which are really needed in order to check the antimicrobial drug resistance or having the, you see, the pure growth of the microorganism. The colonies, they are colorless. They are the non-lactose fermenters on meconic agar and on the EMB agar. Vulgaris and mirabilis, they produce H2S. That is the, on the triple sugar RNA agar, they have the black butt. But none of the other species like Morgani, they do not produce the H2S. The mirabilis, it is indole negative. The other all species of the Proteus, they are indole positive. So all four species, they are urea positive. So you can really differentiate based on to the biochemical test, particularly the indole test for the differentiation of these various species of the Proteus. Now here you can see the swarming overgrowth, and you can really see what is how it happens. You see there are swarming growth, and you cannot really have the isolated colonies. So this is the blood agar, this is the nutrient agar. You can really very well appreciate the consistent growth of this Proteus. So if you see these kinds of growth pattern, swarming, growth on the blood agar or onto the nutrient agar, this is almost characteristic, and we can certainly determine the organism involved in this kind of infection. The treatment is very simple. Most of the organisms, they are very much sensitive to the monoglycoside and trimethoprim sulfamethoxazole. Antibody sensitivity should be performed in some cases where you see you are not getting good response with these antimicrobial drugs like monoglycoside and trimethoprim. Proteus mirabilis, this is sensitive to ampicillin. Again, you see the indole positive species, which are the vulgaris and mirabilis, they are resistant to this sphaerosporin, whereas the indole negative, they are not resistant. There's no specific preventive measures. Hospital-acquired UTI prevented by the removal of the catheter is the only way. So whenever you see there is an infection going on, and what we can do that we can really withdraw the catheters, or we can change the space of the catheters for that purpose. Now we have another two organisms belongs to the genus Bacteroides and Prevotella. The disease caused by the Bacteroides, as we all know, they're the most common cause of serious anaerobic infection. in the gram-negative rods, the Bacteroides and Prevotella are the only organisms which are anaerobic in nature, and they cause sepsis, peritonitis, abscess. And the important pathogens of the Bacteroides are the Bacteroides fragilis, and in the Prevotella we have the Prevotella melanogenica. In some books you will see the melanogenesis, or Prevotella melanogenica is the organism involved in the disease process of the melanogenica. It formerly known as the Bacteroides melanogenesis, and the both still are encountered. So we have the, you see, again renamed for the Prevotella melanogenica as a Proteus, the Bacteroides melanogenica. Important properties of this group of microorganisms: they are anaerobic, they are non-spore forming. Now, the anaerobic spore forming gram-positive rods includes Clostridia. Anaerobic non-spore forming gram-positive rods, they include the Bacteroides. They are the gram-negative rods. Fragilis and Corydons are the human pathogen case of Bacteroides. The Fragilis group is really dominating in the human colon, and a huge burden in the billions of cells in the 1 gram of feces, and found in the vagina of almost 60% women. So the women are the carrier. It is widely found in heavy numbers in the fecal material, and this predisposes to the uterine infection. The melanogenica and Corydons, primarily in the oral cavity, can also be seen. The pathogenesis and epidemiology, as we know, that's a normal flora. So all normal flora, they can have the endogenous infection. The individual can have the endogenous infection. The source is from within the body. And the organisms are the normal flora. They're already in the body. So you're getting infection from the organism which is already in your body. So these kind of infections are known as endogenous infections. And these infections arise from the breakage in the mucosal surfaces whenever there is a damage inside. The diseases, these are not communicable. Local abscesses at mucosal break can also take. Metastatic abscess through the metagenic spread to the area far from the primary lesions is very common. Other organs like lung abscesses by the aspiration of the oral flora can also be seen. The predisposing factors are the surgery, some kind of trauma, chronic diseases, local tissue necrosis, impaired blood supply, and growth of facultative anaerobes. They really predisposes these areas to the infection with Bacteroides or Proteus. The facultative anaerobes, they reduce the oxygen level and allow the anaerobic to grow and both be treated. You see the presence of the facultative anaerobes, they enhances the chances of infection and replication of the microorganism because of consuming the oxygen supply. So the oxygen is depleted to the extent that really facilitates the growth of the anaerobic microorganism. So if you have to go for the treatment of the anaerobes, we will have to consider the treatment of the facultative anaerobes also simultaneously. Because if we kill the anaerobes, but we do not kill the facultative anaerobes, it will not go to work effectively. So we'll have to take into consideration both the anaerobes and the facultative anaerobes that may be involved in that area. It's a capsule-forming microorganism, and the capsule is very important virulent factors in case of fragilis. Other enzymes like hyaluronidase, collagenase, and phospholipase, they also contribute towards the tissue damage. And this is gone true in case of Streptococcus aureus, Streptococcus, we have again these the hyaluronidase, cholineases, and lathistases. But the symptoms of Bacteroides species sepsis, they resemble with the sepsis which is caused by the bacteria with endotoxins, and there is no exotoxin involved. Clinical findings, this group is associated with the intra-abdominal infection, particularly the peritonitis and the localized abscess, the pelvic abscess, and necrotizing fasciitis and bacteremia. Very notorious microorganism, particularly in the abdominal surgical procedures. Peritonitis is very common, it's very severe, it may be lethal sometimes. Apart from the peritonitis, there is local abscesses, pelvic area is involved, and fasciitis. Abscess of the mouth and the pharynx, brains, and lungs. It is commonly caused by the Prevotella melanogenica. because it's the normal flora in the oral cavity. About 20% of the lung abscesses, they are caused by the Bacteroides fragilis. And the diseases, they cause a disease below the diaphragm, whereas the Melanogenica, they cause a disease above the diaphragm. So both species, they cause disease in the thoracic and abdominal cavity. Above the diaphragm, the Melanogenica, below the diaphragm is the fragilis. There is another species known as Prevotella intermedia. It causes gingivitis, periodontitis, and dental abscesses as the organism is commensal in the oral cavity. Laboratory diagnosis for these infections, as it is anaerobic, so we will have to isolate the organism in an anaerobic environment from the blood agate containing kanamycin and vancomycin to inhibit the unwanted organism. Identification of this microorganism is based on the chemical reactions, particularly the sugar fermentation test. There is production of some kinds of organic acids, and we can determine these fermentations are the production of these esters with the help of gas chromatography. And Melanogenica, it produces black colonies. So treatment is, again, you see, difficult to treat these anaerobic gram-positive, gram-negative rods. The fragilis group, it is resistant to penicillin. This group is also resistant to the first-generation cephalosporin, aminoglycoside, the most resistant anaerobic bacteria, and the penicillin resistant due to beta-lactam. So you see, a number of drugs, they are not effective against fragilis. So Bacteroides, they are very, very resistant microorganisms. Do not respond to penicillin, do not respond to first-generation cephalosporin, do not respond to cephalosporin. So the only way left is that we will have to go for the antimicrobial sensitivity testing. Metronidazole is the drug of choice for these anaerobes. And we can combine these drugs with the cefoxitin, clindamycin, chloramphenicol. They can be used as an alternative. Aminoglycosides, they can be combined to treat the facultative gram-negative warts in the mixed infection. Metronidazole and clindamycin, they are the drug of choices for the Prevotella melanogenica. If there is abscess formation, we will have to go for the surgical drainage of that abscess, and we will have to continue accompany with the antibiotic therapy. Lung abscess often heal without drainage with the antimicrobial therapy. This was all about the Bacteroides and Prevotella. We have covered the gram-negative rods, which causes infection outside the enteric tract. Now, we will start in the next lecture with the rest of the members of the family Pteropacteriaceae. Thank you very much.